You can go ahead.
All right, cool. Then good morning, everyone. Hi. Thanks for joining. Thanks for being here. This is the final lecture of the Bayesian Information Processing and Machine Learning course. My name is Magnus. I am a PhD student in Bert's lab. Some of you will probably have seen me sort of sporadically hanging around the class forums, helping people debug their Julia setups and whatnot. Otherwise, I've mostly done behind the scenes work to make sure that the class is running as smoothly as we can get it to. Yeah, besides that, I'm a PhD. I do research related to what we're going to talk about today. And my background is slightly different to most of you. I come from a computational and cognitive neuroscience background. So I know stuff about brains and how to model brains, which you're going to see pop up on different occasions throughout the talk today. Also, I generally like to run a very interactive classroom because the more feedback I can get from you guys, the more I can tailor what I'm doing to what you need, and the more fun it is for me. So throughout, I'm going to be asking you questions on occasion, and I want you to chime in with the answers in the chat room, and hopefully we can get some sort of interaction going. Right. So with that out of the way, let's get into it. So what are we going to talk about today? Because we have this fantastic title for the lecture, What is Life? With the subtitle, The Free Energy Principle and Active Inference. And what I want to try and do today is give you a little bit of a taste of what you can actually do with the tools that you've learned in this class. So far, the vast majority of what we've been doing has been done in the context of processing data. Right? We've learned how to work with various probability distributions and how to update our beliefs as we get data. We've tried our hands at programming exercises where we are modeling different data sets, trying to do different things. We've talked about model architectures and how you can deal with discrete data and continuous data and how you can combine these. And basically, we've looked at a lot of ways you can use all these tools to deal with data sets. And there's nothing wrong with that. Most applications you're going to come across will probably involve a client having a data set of some description and you having to process it. But the tools that you've learned in this class and the concepts that we've been talking about are actually very, very powerful. There's a lot you can do with these, and there's a lot more you can do with them than just process data. And what I'm going to try and do today is sell you on some of the ideas, some of the ways people are applying the tools that you have learned to understand what looks like very, very complicated phenomena. So that also means that I'm not going to be introducing anything new. All the concepts we're going to talk about, it will all be stuff that you have already seen at one point or another during the course. What we are going to do is we're going to try and take those ideas and put a slightly different spin on it. We're going to attach a little bit of different semantics. We're going to attach some different interpretations. And we're going to try and use that to see how much power is actually in here, how far you can actually push the ideas that you've worked with. By the end of it, I hope to have a somewhat deflationary stab at a possible answer to the question we state here. We're hopefully going to arrive at a way we can think about living systems using some of these tools. So this is the battle plan. This is all the stuff that I want to go over today. We're going to have a pretty traditional lecture format. So in the first half of the talk, I'm going to start with the premise for everything. That is, there are some ideas and some concepts and some intuitions that I want to get into your head before we sort of get into the meat of everything. From there, we're going to look at the concept of non-equilibrium steady states and biology, because we want to talk about living systems. That's in the title. And from there, we're going to sort of try and loop back around and get back to something Bayesian, because that's really the goal of everything. And once we're there, once we've sort of established this Bayesian relation, we're going to look, take a brief excursion and look at the concept of a Markov blanket, which is going to be a central concept going forward to the second half of the talk. Then, depending on time, halfway through, we'll have a 10 to 15 minute break and maybe do a bit of Q&A. And then in the second half, we're going to try and look at ways people are using these ideas to think about the world and concepts in the world. First, we're going to look at one of my all-time favorite ideas, the Bayesian brain hypothesis, which is kind of cool. Then we're going to revisit the construct of the free energy, and we're going to try and do some slightly different things to it and see what we can get out there. Then we're going to look at some sample structures, i.e. we're going to look at how we can think about models of the kind of systems we're interested in. And then finally, I have put this goes to 11, and there's props for anybody who gets that reference. So towards the end, I'm going to try and sort of crank these ideas even further and see just how far we can go. So by the end of it, I'm going to hit you with a sort of flurry of concepts and ideas to try and get your mind spinning and thinking about what you can do and what people are doing with these tools. That's if there's some time left at the end. Hopefully we'll make it there. So everything we're going to be talking about today is based on the work of this handsome gentleman that you see on screen right now. This is Carl Friston, who I know you've met before.
We can't see the PowerPoint.
Oh, you can't see the PowerPoint. Well, gosh darn it. Then I'm just talking and you're looking at, Pac-Man. That doesn't really make a lot of sense. All right, is it better now?
No, we still see a white screen.
Still see a white screen.
I can't see it though. It doesn't appear to be for everyone, because I can't see the PowerPoint, and I could see it as well, like in the past.
I see it as well. So
okay.
I don't know.
I see a white screen.
You see a white screen. That is.
Magnus, can you try to reshare your screen?
Yeah,
maybe that'll reset it.
Just hang on a bit. Alright. So does everybody see the PowerPoint now?
Yeah.
Nice. And
what the second thing? Oh, now we see Pac-Man.
Yes. So what if I do this? Just hang on a moment. What if I do this? Do you see the PowerPoint?
Yeah.
Okay, perfect.
And the second thing I wanted to ask, is this going to be part of the exam, or is that just like an excursion for like our interest when foreseeing things from a different perspective?
This is not going to be part of the exam. You can relax and lean back and sort of treat this as nothing but, a presentation of some cool ideas.
Okay, thank you.
Sure. All right. So these are the only two, those of you who had a white screen, these are the only two slides you've missed. Right. So to get back into it, the handsome gentleman on the right. So the guy you see on screen is Carl Friston, who I know you've met before. He was in a couple of Bird's classes as well. There's a couple of different pictures you can go with, some being like very high-res, sort of superhero-esque Quantum magazine pictures. I kind of like this one because it makes him look approachable. And from experience, he's also a really nice guy. But don't be fooled. Like, this man is an absolute scientific titan. He is, like, by miles the most cited living neuroscientist. The guy is an absolute beast. Okay. So now we sort of sketched out what I'd like to do. We can start doing some other things. So where we're going to start is we're going to start with an observation. And that observation is that most systems that you see in the world, if you just leave them alone, they tend to dissipate. That is, if I take a drop of color and I drop it into a body of water, it'll diffuse and it'll dissipate until we have this uniform distribution of everything. Same thing if you look at a lightning bolt. You'll have a snap of self-organization into this impressive, powerful structure, and then nothing. The clouds are the same, and I'm sure you can think of a laundry list of examples if you want, stars and whatnot. But what I want to get at is most systems, if we leave them alone, tend to over time settle into a stable equilibrium with their environment. Those of you who are physicists will be very well familiar with this. The rest of you probably also. But the thing I want to start with is, again, things, if we leave them alone, tend to settle. Now, one of the systems we can think of, sort of as an example of this, is a pendulum. And here you get to enjoy my my fantastic drawing skills in PowerPoint. Don't judge me for the artistic expressions, judge me for the concepts. But if you have a pendulum and you leave it alone, after a period of time, it'll eventually settle into stable equilibrium where it's pointing straight down. And that's doesn't really matter what you do to it beforehand. If you can push it around, you can wiggle it, you can do all sorts of stuff to it. But eventually, if you leave it alone, it's going to settle into a stable equilibrium with this environment where it's pointing straight down. I know there's also, you can have it pointing exactly straight up, and you'll also get something stable. But just for the sake of argument, let's go with the pendulum hanging straight down. And this is an example of a system that just settles into an equilibrium steady state. That's pretty straightforward. But there are other things we could want for this pendulum. There are other things we could want to have it do. Like, for instance, we might want to have it settle at an angle. There's nothing wrong with it. We could want that for various different reasons. So how would we do that? What would we have to do to the pendulum to get it to settle at an angle? This is one of the occasions where I'd like to get some feedback and some input from you guys. So if you can pipe up with your suggestions. Really, this is not a trick question at all. I'm not sure I get notification pop-ups here. All right, so I'm just gonna have to swap back and forth a little bit. You can forcibly hold it there. You can balance it with counter forces. Apply a force opposing the down movement. Yes. So you have something that keeps it in place. But I think your mic is still on. Make it flow partly in water. Oh, nice. I haven't heard that one. That's cool. So basically, what you're all sort of getting at is that we somehow need to interfere with this pendulum somehow. We need to apply some sort of force or put in place some sort of structure to sort of keep it away from its equilibrium. Now, if we do that, we can actually get the pendulum to settle. That means if we have an open system, a system that can interact with its environment, as in we can have forces applied to it, for instance, we can actually have that system settle at a steady state that is not at equilibrium. We can get to a non-equilibrium steady state. Now, why am I spending all this time on pendulums and steady states and all this slightly esoteric stuff? I'm doing that because I want to get back to biology. And one of the things you can observe is that if you look straight down, you will see a biological system. Your view is going to be slightly different from mine because you're a different biological system, but the point still stands. And that system exists very, very far from equilibrium. If you think about it, you are made up of stuff. And there's an almost infinite number of configurations that this stuff could be in. And a very, very small subset of them are consistent with you being you. And yet here you are. So in order for you to keep existing, you somehow have to maintain this non-equilibrium steady state over all of the stuff that makes up you. So we can think of, in some ways, biological systems as systems that persist over time in a non-equilibrium steady state configuration. We can finesse that a little more. So one of the things we could think about is that in order to maintain your non-equilibrium steady state, that is to maintain the configuration that is consistent with you being you and staying alive, there is a set of variables that you need to keep within pretty tightly defined bounds. This is stuff like your body temperature, which you need to keep at 37 degrees. It is stuff like your blood glucose levels. You need to keep them within certain bounds so you have energy to fuel all the other processes. Stuff like your blood pressure, which also cannot be either too high or too low. There's a whole host of different variables that you, as a system, in order to stay alive, need to keep within very tightly defined bounds. And as long as you do that by interacting with your environment, you get to stay alive. You get to maintain your non-equilibrium steady state. Now, under a blanket term in biology, this is nothing new. This is just what we call homeostasis. It's a pretty straightforward concept. All right, so again, why are we spending all this time on this? Why are we talking about homeostasis all of a sudden and non-equilibrium steady state systems in biology? Because after all, this is supposed to be about Bayes. We're supposed to be talking about Bayesian information processing here. And the way we're going to try and get back to Bayes is by an example. So if we roll with the body temperature example from the last slide, right, we all know the body temperature is supposed to be at 37 degrees. So let's try and roll through a little bit of an exercise here. What do you think would happen to a person if her body temperature spiked to 50 degrees? I'm going to go back here and look at your suggestions. She dies. They die. He will die. Yeah, you're all pretty spot on. Spontaneous combustion, I think that requires a little bit more, a little bit of a higher degree, maybe something else. Protein denaturation, nice. But basically, you're all sort of getting on that something would happen to that person, whether they would combust or not, that would cause them to cease living. They would not be able to maintain their non-equilibrium steady state, and they would start to dissipate, whether that is in terms of protein maturation or just processes shutting down or whatnot. System can try to cool down by feedback. Oh, nice. Keep that in mind, because we're going to get to that a little bit later. This is keep this point in mind. All right, just for the sake of example here. What do you think would happen if we take the same person and instead of 50 degrees, we spike their body temperature to 40? Hopefully not spontaneous combustion. Fever? Just going to give a few more minutes for other people to chime in if they want. Impaired bodily functions. All right. And there's still a chance to get the temperature down to 37 degrees without too much damage. You guys are preempting me by like 20, 30 minutes here. It's great. So basically, yeah, sweating. Basically what we're getting at here is if you're at 40 degrees, you have a fever. You're probably not doing too hot. Am I muted? Can you hear me? All right, cool. Yeah, so basically what we're getting at is if you get a fever, excellent, thanks. If you get a fever, you're not really doing too well. Like 40 degrees is pretty bad fever. You're feeling sick, things are not working properly, your body is in a state of disarray. But you're not necessarily dead, though. I know. Whenever I have to switch back to watch the chat, somehow you get to watch that as well. So we'll be back to the slides in a minute. Right. So if we're at 40 degrees, we have a fever, we're probably not doing too well. Now, what happens if we keep going? Now, I think you can start to see where I'm going with this now. What happens to the same person if we set their body temperature at 38 degrees? She feels better. Slightly uncomfortable. Slight fever, still function properly. Still sick, only lightly. Yes, this is bang on. Body defense work less effectively. Yeah, that's probably true, even for all the way up to spontaneous combustion. But basically, most of the time, that's not life-threatening. Exactly. I can't see a first name, but whoever's last name is Okal, which I'm probably also pronouncing wrong, hits it bang on the head. If you get to 37 degrees, that's not really life-threatening anymore. It's very light fever. You're probably not going to be on top of your game. It's not going to be your best day. But it's a far cry from what happens if you're at 40, 42, or even 50. You start to feel better as you get closer and closer. So what if we keep going? What if we get to 37.1 degrees? Uh, 37.1. You probably don't notice that at all. Might just as well be normal. Might not differ from the previous. I get some thumbs up within bounds or normal. You don't notice within a standard deviation, normal functioning. All right. So this is the point I want to get at. Now we're going to go back to the slides. If you're 37.1, you're probably fine, right? I think that's well within the bounds of normal. That means even though we know body temperature is supposed to be 37 degrees, we actually have a little bit of give and take here. There's various reasons why you might find parts of your body warmer or colder. I say when you're closer to the skin, for instance, things might be slightly colder than when it's deep in your core. The blood flowing back through the veins has a different temperature that are flowing out from the heart. You might enter into a sauna and things might change. You might eat a lot of ice cream. I don't know. There's various different reasons why your body temperature might vary, why there's a little bit of wiggle room around the set point of 37. And all of that is within normal bounds. All of that is fine. But a number with a bit of wiggle room, a set point with a bit of wiggle room, that's we know what that is. That's a distribution, right? So actually we can think of body temperature as having a distribution, right? I put here a target prior distribution for reasons that become slightly clear later. But basically we can think of you as a biological system having a prior distribution over a desired set of states that you want to occupy. But you can wiggle a bit, as long as you're within those bounds, you're going to be fine. You're going to be able to maintain your non-equilibrium steady state. You're going to keep your homeostatic variables under control. And you're going to be able to stay alive. So we can think of these non-equilibrium steady states as a set of priors over a set of relevant variables that are mostly homeostatic. But that's not all. Like, you are, after all, an open system that is moving around the world, and you're doing things and you're interacting with your environment, and a lot of stuff is happening. You're an open system, just as we looked at earlier. And that means a set of prior distributions is not going to be enough. We're going to have to introduce a posterior as well. This is going to be the posterior over your actual body temperature after you have interacted with the world. So while your prior, your goal state is going to be this tight distribution over 37 degrees, things might happen that make you slightly warmer or slightly colder. And you need to sort of keep track of the actual state of your system as you are out and about in the world. But now we have a target prior, and we have a posterior after interacting with the world. And we can think of the task of the system as lining these two guys up. So we have a P and a Q, and we want to get them to match. And this is a kind of problem that we know how to solve, right? This is a Bayesian inference problem. This is a variational inference problem. This is about matching up two distributions. And these are the kinds of things that we have dealt with in this course. This is the kind of stuff that you know how to do now. And so just to recap very, very briefly, what we've done so far is we've started with the observation that most things, if you leave them alone, will tend to dissipate and go to equilibrium steady states. Then we've seen that we can also think of systems that settle far away from equilibrium, given that they are open systems, and we can think of biological systems in this way. And now that we've sort of established that we can think of biological systems as non-equilibrium steady state systems that occupy some adaptive set of states that we call homeostasis, we can define, sort of finesse that a bit, by saying the adaptive set of states are going to be given as a set of target priors. And the task of the system, the task of the agent, the task of the organism is to match up its posterior after interacting with the world with its target priors. So we're moving at quite a brisk pace here. But I hope that this makes sense to everybody. Everybody still on board? Get a yup. I get 2 yups. I get 2 yups and a yes. Actually, maybe that is actually a way for me to get notifications when we do this. All right, screw it. We're just gonna have to move back and forth. All right. So this already tells us something about what we can do with the tools that you've learned. Because notice we haven't introduced anything new really here. We've hardly looked at any equations. This is the closest we've gotten so far, where we have a P and a Q on screen. But this is all within the bounds of the tools for Bayesian information processing that you have learned and that you have studied. And we can apply these tools to think of biological systems in this way. So already here we're moving quite far into sort of things that illustrate the power of going full Bayesian. Because again, this is a lot more than just processing data. Right. So the next thing we're going to do is we are going to talk a little bit about what kinds of systems we would want to think of in this way. And we are going to do that by introducing the concept of a Markov blanket. So there's several different characteristics we could pick out, but we're going to focus on that of the Markov blanket. And a Markov blanket is a graph theoretical construct. And it basically denotes on a graph a set of nodes that, if known, would render 2 other sets independent. So that means everything here on the dashed line, I don't know whether you see my cursor, but I'm moving it around the dashed line. Everything on the dashed line would constitute a Markov blanket separating the internal states, the S's, from everything else, i.e. the giant R on the left. Simply means that if we know everything on the dashed line, then all the S's and the giant R are conditionally independent. So if we pull that back a little bit and think of this in terms of biology again, this simply means that in order for us to be able to talk about a system in this way, we have to be able to separate the system from everything else. There has to be some sort of a boundary that we can draw and say, this is the thing we're talking about, and everything else is not that thing. That's a pretty reasonable assumption. In your case, for instance, we might draw your Markov blanket at the skin, because that is a pretty good barrier between you and everything else. Now, on the contrary, if we don't have a Markov blanket, if we're not able to separate the system from everything else, we're back to this situation of diffusion and gases and solvents and whatnot. So we're going to focus on systems that have a Markov blanket. All right. So if everybody sees the Markov blanket here, the Markov blanket that sort of insulates the agent from the environment, i.e. the thing that is within the dashed line from the giant R. Oh, and I got dragged here back for some reason. Right. So everybody sees the Markov blanket, right?
Your shared screen is gone, so now we just see your face.
God damn it. This is a lot more difficult than it has any right to be. How about now?
Now we're back.
Perfect. So does everybody see the Markov blanket? I'm going to switch back again because I can't see your chats right now. I see a flurry of yeses, which is perfect. All right, cool. Because if we have a Markov blanket, this is one way of writing it. You've seen this figure before because I have blatantly stolen it from Bert's lecture on intelligent agents. If we have a Markov blanket, this is one way of writing it, but we can also write it like this. Now, what I've done here is that I have carved up the entire world into 4 sets of states. We're going to have the internal states of the agent. So that's everything inside the Markov blanket, which is going to be mu on the right. We're going to have the external states of the agent. So that means the world and everything in it on the left, which is going to be eta. And then we're going to have the Markov blanket between the two, which is going to be the remaining 2 nodes. And here in my slightly hacky graph, the arrows are going to denote dependency relations. So what we're going to do to the Markov blanket now is that we're going to carve it into 2 sets of states. First of these are going to be sensory states, which we're going to denote S. We're going to define these in terms of their dependency relations. So sensory states are influenced by external states, but do not influence external states. And to make that a little more concrete, sensory states are the parts of the boundary that the system can use to take in information from the outside world. So in your case, it would be the little cells on your retina that are picking up light. It would be the little hairs in your ears that are vibrating as you're listening to me. The receptors all over your skin that sense touch. All the things that you use to take in information from the outside world. We're just going to lump it all together. We're going to put it in a block called sensory states. The other part of the Markov blanket, we're going to deem active states. And these work the other way. So active states are states that influence the outside world and are influenced by internal states. So now we're back to the points that I sort of asked you to latch on to earlier. These are states that govern things like secreting sweat or pheromones. These would govern things like muscle contractions. These are all the things that you as an organism, as a system, can do to influence the world around you. Now, this also foreshadows a bit where we're going with this, because this also immediately tells us that there are 2 ways for the system to make sure that it stays within its homeostatic target priors. That is, either we can update our beliefs, we can go through the sensory states and influence the internal states and make sure that our beliefs match what we want to be. Or we can go the other way. We can go through active states and influence the external world such that the data is now in line with what we want them to be. So the difference here is the difference between adjusting your expectation of how cold or warm you're going to feel and putting on a jacket. One is adjusting your internal model by moving through the sensory states. And the other one is intervening in the generative process. That is, you are actively doing something in the external world. And we can use both of these to make sure that our P's and our Q's line up. Now, there's another reason that I'm showing you this figure. And that is because it gives us a clue as to where on a graph we would want to put our target priors. Because so far, I'm doing this very, very abstractly. I'm talking about a distribution over body temperature. I haven't really committed to anywhere putting it into a graph or into math anywhere. I'm just stating that it's there. But if we look at this graph, we can see that this place that makes most sense to put this prior, to put this target prior, would be in the sensory states. Because by definition, the only way for the external world to influence the internal milieu of the organism is to go through the sensory states. So that means if we put our prior over the sensory states, that is, we specify a set of desired observations, we'll have control over everything that the external world can do to influence our internal states. Or to put this a little bit more concretely, what you care about as an organism is not necessarily maintaining your body temperature at 37 degrees. I mean, you do, but as a proxy for that, you're going to care about not feeling cold. You're going to have a preference for sensations of a particular kind, which are going to be not too cold, not too hot, but just right. And you're going to act on the world in a way that you believe will bring about those desired sensations. So if you go outside and it's cold, you're going to put on a jacket because it'll cause you to feel not too warm, not too cold, but just right. All right, let me see. Yeah, so there's one more point that I wanted to hit before we do a break, and I'm not sure how we're doing for time because I can't even see my timer right now. But
11:30.
11:30. Oh, good. Then we're almost bang on. Thank you. So the last thing that I want to take a look at is, where do these priors come from? Because right now I'm just pulling priors out of thin air. I mean, I picked body temperature at 37 degrees because we know something about that from biology. But where would that 37-degree temperature come from in the first place? The argument here is sort of an argument by contradiction. And it goes something like this. So any system that exists, any organism you can think of, has to have a set of adaptive priors. And that is so because if it did not have a set of adaptive priors, it would not be able to maintain this non-equilibrium steady state. Again, because if you think back to our example of somebody's body temperature spiking to 50 degrees, if we have a version of the human being that has a target prior set at 50 degrees instead of at 37, that species is not going to survive for very long. So sort of by contradiction, we can say that any system that exists must have a set of adaptive priors. There must be a non-equilibrium steady state that describes this system, this kind of system. And then by evolution, any system that exists has to have evolved an adaptive set of priors. Let me see, was there anything that I didn't cover? Yeah, so the last thing that we sort of briefly going to tag on is that we are going to equate maintaining a non-equilibrium steady state. I should have said this earlier. We're going to equate maintaining a non-equilibrium steady state with maintaining your Markov blanket. That is, as long as you are able to maintain your structural integrity and your separateness from everything else, we're going to say that is you maintaining a non-equilibrium steady state. Because if you don't, you will start to dissipate and we will lose the ability to distinguish between you and the world around you. And then it's a fair point to say that you are no longer existing as a separate thing in the world. So that is everything that I wanted to cover in the first half of the talk. So if we are about 11:30, I'd say we can do a 15-minute break, refuel on some coffee. That's what I'm going to do. And then I'll hang around for a brief Q&A. I can see you back at 11:45. And then, yeah, in the meantime, if there are any points that are unclear, if you have any questions, just throw them at me and I'll do my best to clarify. Otherwise, see you at 1:45.
I did actually have a question for you.
Sure.
About the graph. You basically have an adaptive prior, but wouldn't that be before you are in your sensing state, basically?
What do you mean you are in your sensing state here?
Or, well, you basically, if you have the graph, you said that the prior is placed basically in your sensing state.
Mm-hmm.
But why would it be there, as the prior, technically speaking, has nothing to do with your sensing state, right?
We place it there because if we condition on a prior the sensory state, that renders the external and the internal states conditionally independent. I.e., there's no way for the external world to influence the internal state of the organism. That's the same as saying, if I know everything that is happening at your skin all the way around, and I know everything there's to know about that, then knowing the temperature, say a centimeter away from your skin, is not going to tell me anything new about the states inside of your body.
Okay.
Does that make sense?
Sort of.
So it's basically, because we define sensory states through the dependency relations, conditioning on sensory states renders the outside world and everything inside the agent independent.
Okay.
And that's why it's a good place to put our adaptive priors. The fact that it also has sort of a nice interpretation attached to it in terms of you acting on your observations is just a nice bonus.
Okay. Great. Thank you.
Thank you.
Magnus, do you want to keep the recording on, or shall I stop it and then start again later?
I think we should keep it on just in case there are questions.
Okay.
Because having those in the recording, I think, would also be nice.
All right.
And then you all get to enjoy my fantastic looking mug in the meantime. We can stop the screen share temporarily, though. All right, so there's a question. How are the priors present in our body as a system? Are they somewhere in our minds? That is a really good question. So one way to think of this is that your body and your mind and everything that is you is a model of the environment that you're in. What do I mean by that? If you look at, say, a fish, the sort of physical makeup of a fish tells you a lot about the environment that the fish lives in. Like it wouldn't survive on land, for instance. So in the same way, we can think of systems, if we really commit to this, as being models of the environment that they're in. So the priors would be, yeah, just part of your system. Now, exactly where they're located in terms of, is it like a metaphysical, psychological thing? Most likely not, but we don't really know. And how exactly it is encoded in your biology probably also varies from species to species. So that is the long answer. The short answer is we really don't have much of a good idea, but it's a good way of thinking about things. People have been doing this in biology for a long while in terms of set point theories and whatnot. One thing we could ask in that vein is whether the priors are always all fixed. And sometimes there's evidence that some priors would actually have a bit of give and take as well, i.e. weight, like body weight, can often be studied using a set point. And what you get is if you gain a lot of weight, you might actually be able to move, or lose a lot of weight, you might be able to move your set point around, and then it makes it hard to deviate too much from that set point. So yeah, the long-winded answer is we think of the system as a model of its environment. The shorter answer is that's really tough to answer. I hope that clears something up. All right, cool. If you understand, then please write a paper. Now, the relation between how these things map onto things like cognition and psychology and whatnot are still very much open questions. There's a lot of interesting work being done in that vein these days. So there's an opening to get into it if you want. All right. I think we have also hit quarter 2, so I'm going to pick up the second half of the presentation. Which means that you should now be seeing, sorry. All right, everything works. Just shoot me a flurry of messages if you don't see the presentation. So hi everybody, welcome back from the break. I hope you've had some time to mull all of this over. So to pick up the sort of second half of what we're going to talk about. Now, in the first half, we sort of established a way of thinking about biological systems using the tools of Bayesian and variational inference. And in the second half, we're going to take those tools, sort of things we've outlined, we're going to look at different things people are doing with them, different things you can do with them. We're going to look at the Bayesian brain. We're going to revisit the free energy. We're going to look at some sample structures for the kinds of systems one would want to think about. And finally, we're going to go to 11. So all of this, again, is done under the heading of the free energy principle, is what it's called. And that sort of describes this way of thinking. So let's get into it. So we're going to start by the Bayesian brain. And the Bayesian brain originally originates, let's put it as a purpose, it originates with this handsome gentleman, whose name I am going to butcher now. He is Hermann von Helmholtz. If any of you are German, you can probably pronounce it better than I am. And he was a brilliant scientist active in the mid to late 1800s. And he did a lot of things. But one of the things that he did is he started this idea of the brain as a prediction engine. To paraphrase, he said something like, perception is inference about the causes that would have to be present, such as to produce the inputs on our sensorium, something like that. Basically, he said that perception is the process of inferring the hidden state of the world that is most likely to cause the data that is impacting your sensory states or your sensorium. So, for instance, right now, photons are hitting the cells in your retina. And there you could have many interpretations, but most of you are going to be passing them as coming from a screen that is in front of you, and on that screen is a slideshow with a picture of Helmholtz. At the same time, you have sound waves hitting the cells in your ears, and you are inferring that the most likely explanation for these sound waves is that there is a guy who looks and sounds roughly like me, who is talking. You're going to combine these sensations and infer that the most likely state of the world right now is that you are in a lecture, it's online, and there's a guy who looks and sounds roughly like me, who is doing a presentation. Now, there are other possible explanations for this. Like you could be a brain in a jar, and somebody could be sending electrical impulses directly to your nervous system to recreate all of this. But the most likely one is that you're in a lecture and that I'm talking. So we can think of, and that's what the Bayesian brain hypothesis is, we can think of the brain as an organ that does predictions and inference about the world. Basically, the purpose of this thing that we carry around inside our head is to be a prediction engine. It is to make a model of the outside world and encode it in neuronal connections and firing patterns and all the fantastically complicated and interesting things that the brain does. But all of that is done in service of predicting the world around you. If we sort of roll with this, then that is what the brain is for. It is a prediction machine that tries to make sense of all the sensory inputs that you're getting. So if we roll a little bit with this idea, people have actually tested it. And what we're going to look at now is probably some of the most famous single neuron recordings ever done. I don't know, do any of you dabble in neuroscience at all? Nope. All right. And there's somebody complimenting me on pronunciation. Oh, thank you. I've tried really hard. Oh, we actually have some people looking at neuroscience, and are not yet. I like the not yet. All right. So those of you who go into neuroscience and want to sort of sniff around this area, especially if you venture into the sort of computational and decision-making neurosciences, you're very likely to see these slides again. I was at a conference once and I saw the same single neuron recordings presented like 5 times by different speakers, because they are just, it is such a good experiment. It is such a clear setup. It is really like exemplary science, right? So without further ado, let's bring them up. So what are we looking at here? Well, let's skip the point here. Right, yeah. So, yeah, so I skipped a point. I'm just going to roll back a little bit. Helmholtz, as I am and as other people are, was interested in studying the kind of things that go on inside the brain. He came up with this idea of the Bayesian brain, but he couldn't test it. Because back in his day, like neuroimaging tools weren't that advanced. Like having to look at a brain would mean literally having to find a skull, open it up, and take a look at the brain. That's not really conducive to figuring out the kinds of computations that go on inside. Fortunately, we've gotten a bit better since then, and we can do these single neuron recordings now. We can do fMRI, and we have all these nice tools for looking at the computations inside the brain. And the single neuron recordings that we're going to be looking at here are going to be from the dopaminergic system of a rat. So basically what these guys did is they inserted tiny electrodes inside the brain of the rat while it was still alive. And from these electrodes, they can measure the action potential, so the spikes sent out by neurons. So whenever a neuron in the vicinity of the electron will spike, they'll get a small signal that they can pick up. So what we're looking at here, I don't know if you see my cursor, but I'm going to pretend that you do. So what we're looking at here is recordings from the dopaminergic neurons of a rat. And on the x-axis, we have time. And then each row is going to be recordings from a single electrode. So each little dot will signify an action potential. So that is a neuron firing. And this sort of skyline looking plot at the top is just the aggregate of all the black dots. So what this rat is doing is, it's a rat in a cage. It's going about its business. It's doing rat things. It's being a rat. But throughout its life in the cage, it has learned that when a light that is installed in the cage flashes, a little while later, it'll get a reward. So inside of the cage where the rat's running around, there's a little light source, and sometimes that light source will flash, and then a little later, it'll get a reward. And by this point, the rat has seen this thousands of times. Even though it is a rat, it has learned how this kind of thing works. And it is well aware of this relation. So if we start from the top here, what we're going to see is at the very left of the top plot, the rat is just going about its business. It's being a rat, it's doing rat things, thinking rat thoughts, being ratty, doing ratty things. And what we have is baseline activity. Now you'll see the sort of sporadic intermittent firing in sort of a pretty much constant stream. And that always happens. Your brain is never quiet. There's always some sort of background activity going on, some processing, some action potentials flowing back and forth. There's always some level of baseline activity. So the rat is going about its ratty business, and then all of a sudden, out of nowhere, a reward appears. So a reward in this case is a food pellet, because rats, like most of us, like food. So rat is just ratting about, and then all of a sudden, a reward, a food pellet, is dispensed to the rat, out of nowhere. And we can see the rat going, oh my God, this is great. And how we see that is immediately following the reward, there's a spike in neural activity. You can see here, there's a lot of firing going on. And then, after the initial burst of joy, the rat goes back to baseline activity. Now, there are a few ways we can interpret this. The most logical, which people have thought for a long while, and which you, if you dabble in economics, you're still going to find a lot of this kind of thinking, is that these neurons would encode value. These are going to encode the value of the reward. So immediately following the reward, we see a spike in the reward system, which tells the rat this is a rewarding stimulus. This is a good thing. This is a nice thing. That's one possible interpretation. But if we roll with the idea of the brain as a prediction engine, there's another interpretation. And that interpretation is that these neurons are encoding surprise. So if you imagine the rat just going about being ready, it has a model inside its Bayesian brain of the environment that it lives in. It's constantly trying to predict everything that's going on, constantly trying to predict its sensory states. And then out of nowhere, this reward gets dispensed. But the rat did not predict this. Well, how could it? So if we take that view, then this spike in activity encodes a spike in surprise, which you know as like Bayesian log evidence or free energy, or however you want to interpret it. If we sort of roll with that explanation, maybe these neurons are encoding prediction errors, they're encoding surprise. We can take a look at the second row. Now the second row, the rat has calmed down again after this momentous occasion, and it is again back to baseline activity. So it is going about its ready business. And then we get the light flashing. This is here marked with a line that says CS for conditioned stimulus. So the light is flashing. And again, we see a spike in activity. That makes sense, right? Suddenly there's light flashing. This is not a stimulus we have predicted. This is surprising. So if we are neurons that encode surprise, we're going to fire. But what happens later? If we go along after it returns to baseline activity, we get the reward dispensed. This is marked here with R and a little black triangle. This is from 97, so we're going to forgive them for not having time on their x-axis explicitly. But when the reward is dispensed, there is no change from baseline. How can this be? Because after all, the stimulus, the reward appearing, just before, just in the plot above, elicited a spike in activity, now has absolutely no effect on the firing rate of these neurons. And that makes sense when we acknowledge that the rat has learned this mapping over thousands of trials already. The rat knows at this point that a little while after the conditioned stimulus, a little while after the light flashes, it'll get a reward. There'll be a food pellet dispensed. So its internal model of the world, its prediction engine, already predicts there's going to be a reward at this point. Therefore, the appearance of the reward is not surprising anymore, and therefore we don't deviate from baseline activity. Already here, this is pretty cool, to me at least, because it shows very explicitly that these neurons are not just encoding the value of a stimulus, because the stimulus is the same in the first and second row, but only in the case where it's not predicted do we get a spike in activity. And so to hammer this point home, we can look at the last row. So at this point, the rat is again back to baseline activity. All is good. Then we get the conditioned stimulus, just as we did in the last row. So we see here, light is flashing, neurons are spiking. And then a little while later, a reward is supposed to appear, but now there's no food pellet dispensed. And what we see is not that the rat stays at baseline activity. Instead, we see a cessation of firing. So these neurons, they're expecting a reward to appear, but nothing does. So they get surprised. We get surprise with the opposite valence. Now it is not something appears that we did not predict. It is the absence of something that we did predict. And that causes a cessation of firing. And again, the important point here is, as far as the environment that the rat is in, nothing is happening. There is no external stimulus to trigger this cessation of firing. to me, that makes a strong case for treating the brain, at least in this part of the brain and this experiment and whatnot, all the usual caveats, but for thinking of the brain as a prediction engine. Because what this set of data very, very clearly shows, to me at least, is that the brain reacts to faulty predictions and not just to external stimuli. So again, to me, this is just a beautiful, beautiful piece of experimental science. It has a clear hypothesis, it has strong theoretical backing, it has a very, very clean and simple and exceptionally effective experimental design with very, very convincing results. Like, this is just beautiful, beautiful science. And if we can ever do anything that is as cool, you should absolutely do it, because this is just an absolute power move. All right. So that is what I have to say about the Bayesian brain. But basically, is this idea that sort of we can interpret again within the free energy principle that the brain is fundamentally a model of the world, and we're trying to make that model match whatever's actually going on. Right. So now we're going to switch gears a little bit, and we're going to look at the variational free energy. Now this guy you've also all seen before, because you've all done variational inference. I'm going to use S for the hidden states, so the latent states of the model. I'm going to use X for observations, and I'm going to condition everything on some control states U. And why I do that will make sense a little bit later, but for now, just trust me that this is something we can do. And as you are very well aware, we can take this and we can split it into an accuracy and a complexity term, as we have done in the lesson on intelligent agents. And now we're going to talk a bit about intelligent agents, because this is all well and good. Like, if you take an agent and you optimize this here, you're going to get an agent that accurately tries to predict its environment. That is the accuracy term. It is trying to do so with the least complex model. That is, it's trying to tweak its priors as little as possible. That's a complexity term. So we have something that is trying to accurately predict the world around us with sort of a built-in Occam's razor, looking for the simplest explanation. And that's pretty nice. That's all well and good. So why am I showing you this? Well, it's because we've been very sort of high-flying so far. We've been way up in the upper levels of abstraction. And I want to show you what this actually looks like if you implement it. What do you get if you take an agent that optimizes this functional and has a goal prior? So we have a biased generative model with a prior over P of X. If you do that, you can get something like this. This is a good friend of mine. This is the Bayesian thermostat. Now the Bayesian thermostat is sort of the hello world problem of this kind of thinking. It is a little agent that lives on a 1D number line, and it can observe its position and its temperature. So if you wanted a mental image, you can imagine a little cart that can move back and forth, and it is moving towards or away from a fire, so a heat source. And we take this little agent, and we give it a goal prior, a target prior, a prior over its observations that it's trying to fulfill, that we put at 4. We can interpret that 4 how I want to be, but just we put it at 4. And then we start the agent off in a decidedly wrong position. So if you look at this plot, we have time on the x-axis, and then we have 2 curves, one denoting the position and one denoting the temperature. So the position being the position the agent is at, and the temperature being what it currently observes. If we start way off to the left, we see that the agent starts by observing a decidedly wrong temperature of about 20, which is way off from the 4 that it is looking for. But as we move along through time, we see that this agent smoothly and accurately navigates to a position that elicits its desired temperature. That is, it settles the blue line here around 5, because that produces the temperature of 4 consistently. And then it remains there. So I'm not showing you this because I want you to be impressed with finding a number in 1D. That's a pretty straightforward task. The key thing here is how it's done. So this agent that we have here, this is an agent that does not have a concept of a reward function. There's no rewards going on here. It also doesn't have a concept of a policy. We're not learning a state action mapping. We're not really doing any of that here. This is an agent that just at any point in time is greedily following gradients of free energy. It's just optimizing this free energy functional. It's doing so in a greedy manner. And if we do that, we can see that it produces these nice aesthetic trajectories that are consistent with sort of long-term purposeful behavior. And the key thing is we can get that without any notion of planning, without any notion of a policy, without any notion of a cost or a reward function. We're simply doing inference as you have already learned. And from that, we can get these types of control systems. And that is pretty cool. So again, to reiterate, the point is not to find a number in 1D. The point is that we can do this without basically any of the trappings that you would need if you wanted to do this with, say, a reinforcement learning type of setup. We're just doing inference as you already know how to do. That is pretty cool. However, this agent also has its shortcomings. Like this agent is a greedy optimizer. It is at any point just following the path of least resistance, the path of least free energy. That's what it's doing. So if we have a really complex task that actually requires long-term planning, this guy is also going to fail. If we want long-term planning in there, we need to introduce the future. So this figure, again, I'm pulling up because it is really nice for illustration purposes, and you are all familiar with every single facet of it. But what I want you to notice here is I want you to pay particular attention to the time indices. So what we have is not just st-1 and st. We actually have t+1 and t+2 and t so on and so forth, up until horizon t plus capital T. That means if we have an agent with a generative model that looks like this, now suddenly we're in a position where we can plan over multiple time steps. Everybody see that, right? We have a series of control signals. Doing inference in this model will give us a series of actions, a series of views that the agent believes will get it to its goal prize. Incidentally, if you remember back to the that's not intelligent agents, we used to put the goal prize on the observations. They're down here in the red squares. The reason we put them on observations harkens back to this idea of priors and sensory states. So what we're actually doing when we make these types of agents is we're hijacking sort of mathematical description of the principles of homeostasis, and we're using that to design intelligent systems. That's pretty cool. So this is one way of dealing with the future. If you make a model that does this, you get into the field of, say, planning as inference and control as inference, and you're going to get a system that will try to look for the shortest path between its current state and the desired goal state. So it is sort of a mostly exploitative agent that is trying to perform a task as quickly and efficiently as possible. But there are other ways we can go about introducing the future. And I'm going to show you one, which is related to some of the research we're also doing in the lab, some of the work that I'm doing. And that is, instead of just adding more nodes to the graph here, I'm going to take the functional and I'm going to take the expectation of it. Because we can say that since all our observations in the future now have to be random variables, they have to, because the future hasn't happened yet, so there's no data. Then, as a good proxy, I can take the expectation of my free energy under this posterior predicted distribution over my future observations. I promise this is about as hairy as it's going to get math-wise. We're going to get back to all the other stuff soon. So basically, you all recognize this guy here as the free energy we had before. And then I'm just taking the expectation under this guy. And if I do that, I can once again split it. I can split it like this. And then I can sort of split the expectation as well. So I get something that looks like this. Now, why would we want to do that? Well, we get two terms out of this. One is a cross entropy that tries to match up our posterior predictive over distributions with a target prior. So here we can put a prior over the kind of observations that we want our agent to have, i.e. that is consistent with the goal that we wanted to achieve. And this term will then try to bring the agent as close as possible to that goal. So it's trying to bring its posterior beliefs in line with the target that we set for it. Now, the other term here approximates a quantity called information gain. And what this does is it puts value for the agent on visiting states that are informative. So an agent that optimizes this term, when this term dominates, is going to visit regions of its state space that it hasn't seen before and that it believes will carry information about the task it is doing. So we're going to have something that actually encourages the agent to go and explore its environment in order to learn, in order to get information. Now, to put a little bit sort of a more down-to-earth kind of nomenclature on it, which I prefer, we get two terms. We get an instrumental value that is a goal-directed, exploitative, sort of greedily pursuing our targets in the world term, which we can deem instrumental value because it is instrumental, it's solving the task. And we get a term that puts a value on information gathering, on learning about the environment that the agent is in, on exploring, which we can deem epistemic value. So if we take this approach instead and optimize this quantity, which we call the expected free energy, what we're going to get is an agent that actively sort of balances exploring its environment and exploiting its environment for maximum gain. I don't know, do any of you dabble in reinforcement learning? There's a bit. All right. Nope. Those of you who are somewhat familiar with reinforcement, a bit, yes. All right, then you probably have heard of this idea before, the exploration-exploitation trade-off. So one of the problems that people are trying to solve in reinforcement learning is how to balance exploring your environment and learning enough about it that you can make good decisions, with exploiting your environment, that is picking the best decisions to maximize your rewards. And there's a bunch of different algorithms, a bunch of different takes on how people go about this. But excuse me, one of the things that we sort of get if we take this expected free energy approach is a very natural resolution of this kind of dilemma, because now both the exploitative and the explorative component are baked into the functional that we're optimizing. And everything here is conditioned on use. So it's conditioned on a policy, on a particular sequence of actions. So we can optimize this by optimizing U, i.e. by optimizing our sequence of actions. So this gives us a nice way of constructing agents that adaptively balance exploration and exploitation, which is pretty cool. So when might you want to do either of these things? Well, you can imagine, for instance, if you're designing a controller for an industrial robot. In that case, using something like this, like the expected free energy, is probably not a good idea. Because after all, if you have this giant machine, giant robot arm swinging around, exploring its state space, that could be dangerous. That could legitimately be a danger to people and other equipment. In that case, you might want to go with something like the extending the graph approach, because that gives you some setup where your robot arm will move in the shortest path and still be able to handle perturbations and whatnot because it's doing inference. Now, on the other hand, if you're in a situation where you want exploration, like let's say you're doing you're mapping out a building, or you are dropping a rover in an unknown environment, or you're doing like aerial surveillance or something, then it might be a good idea to look to using the expected free energy, because then you get this adaptive trade-off between how much the agent explores the space that it's allowed to move in, and how much it is looking for the thing that you want it to look for. So again, this latter approach also falls out of the free energy principle, and from thinking about these terms in a slightly different way. The other thing that I want you to notice is that I haven't used any tricks or any moves or anything that is not already covered by the tools you've done in class. This is just variational free energies you're used to and then taking an expectation. There's really nothing fancy going on here. So what I want to get at with this section is if you want to design agents that are free energy minimizing agents, there are a couple of different ways we can go about it. And each is going to have their pros and cons. And if you want to get into this kind of stuff and try to apply it, then it's going to be up to you to make good informed decisions about what you want to do. All right, let's see what we have here. Yeah, so finally, and I think we've got plenty of time left over. Let me just check. Yeah, we've got enough time to work with. So finally, we're going to try and crank this whole thing up to 11. Because again, there's a lot of stuff we can do with this. And I've given you some ideas. So we've looked at the Bayesian brain. We've looked at how to design intelligent systems that do various things based on these ideas of simulating biological systems using the Bayesian formalism. But we can actually go further. There's even more we can do with this. And we're going to try and crank this up as we go. We're going to look at this figure again. And now we're not going to look at the time indices. Now we're going to focus on the fact that most of these boxes are blank. Right? And you've been putting all sorts of stuff into them over the course of the course. You've had Gaussians, and you've had multinomials and hidden Markov models and what have you. There's a lot of things you can put in here. But we haven't committed to any particular structure really beyond this. And the fact that the nodes are blank mean that we can put anything that we want inside of a node. If we take this node here, for instance, just this one guy, as long as I take care of the edges, that is, as long as I take care of the Markov blanket of the node, I'm free to do whatever I want inside this node. Like I could, for instance, jam an entirely new state space model in there. That's allowed. I can have this whole entire structure just hiding within a single node. And that is perfectly fine. As long as I take care of the edges, as long as I take care of the Markov blanket, then everything is fine. All the math checks out. Now, why would I want to do this? I want to do this because at no point in this entire exposition have we committed to any notion of scale. We haven't talked about spatial scale or temporal scale or anything at all along those lines. Everything is abstract and probability distributions, and time is indexed by time steps. These can be as fast as we want them to be, as slow as we want them to be, and they can be as big as we want them to be, as small as we want them to be, as long as we take care of the edges and as long as we take care of the Markov blanket. So this agent, for instance, here would run inside of one time step of the larger agent, and we can keep going. So if we take a step back, that means that I can think of you as a biological system. You're going to have a Markov blanket. You're going to be interpretable within all these different kinds of formalism and as doing Bayesian inference and what have you. But I can also think of you as being made up of component parts. I can just as well think of you as being made up of a series of organs, for instance, bones and whatnot. Now, each of your organs is also going to have their own Markov blanket. Like I can delineate your spleen from everything around it. And I can figure out what are the sensory states, i.e. where does the spleen interact, get information from its environment, and what are the active states, what does the spleen send out? And we can go one step further, because the spleen is going to be made up of cells. Each of those also going to have their own Markov blanket we can draw at the cell membrane. And they're going to be channels for taking in information, like ion channels, for instance, neurons, and sending out information. And we can keep going onto component parts of the cell and so on and so forth. And as long as we take care of the Markov blanket, then this formalism applies all the way down and all the way up again. We can use the same tools, nothing but the calculus probability theory, to think about all these things. That to me is a really cool idea. There's some real power here. But we can actually go one step further. Right? We can crank this up even one more bit. To do that, we're going to look at this figure again. And so far, we've focused almost exclusively on the right side of this figure. We'll deal with internal states, sensory states, active states, agents and organisms and their Markov blankets. And we've sort of been focused on the agent or the organism as a system that we want to talk about. We haven't really talked about the left side. And the left side represents everything else. But as you'll notice, the Markov blanket is symmetric. The Markov blanket insulates mu from eta, but it also insulates eta from mu. And that means that I can do something like this. So what I've done here is I've taken the same figure, but now I've split it into two agents. Each with their own internal states, each with their own sensory and active states, each with their own Markov blanket. It's just that I can see I've missed something up here. So flip the top and bottom sensory active. But basically, the sensory states of one agent is going to be the active state of one agent is going to be the sensory states of one agent, the other agent, and so on and so forth. So these should be swapped. The active states is the sensory states of this guy. And now we have, without doing anything extra, now we have a formalism for thinking about systems of interacting agents. That is, from my point of view, I am an agent. I am a system. I do things. And all of you out there are part of the external world. However, from your point of view, you are an agent. You do things. You have your Markov blanket to maintain and care about. And I am part of the external world. So now we have this, without doing anything extra, we can model systems of interacting agents with shared Markov blankets, which again starts to get us into how we can think about groups and multi-scale systems and all these sort of very bleeding edge ways people are applying this kind of principles. And that also, like, hopefully throughout this whole exposition, we've been sort of trying to, at least at the end, get at a possible sort of deflationary answer to the question that we asked at the beginning. Is there somewhere in all of this a possible answer hiding for the question, what is life? What can we think about what life is actually? And if we commit to this line of thinking, if we really commit to it, we get a very deflationary and not particularly satisfying answer, which is that life is nothing but Markov blankets all the way down. From the highest level to the lowest level, there's nothing but Markov blankets all the way down. And that is the last point that I wanted to make today. So for those of you who think this is also cool and want to sort of do a bit of dabbling, here are a few resources you can look to get started. So split them into 3 categories, one being a more lighthearted category, which consists of blogs and YouTube videos, then some research papers, and then the Here Be Dragons section. So for the lighthearted section, there's a nice video by Sean Carroll, who is a physicist and an excellent science communicator, called What is the Purpose of Life? He takes a slightly different perspective in terms of entropy, but it's a really nice video and sort of gives some more perspective on how you can think about these things. And translating between his slightly different perspective and this one is pretty straightforward. Then there's a blog post by a guy named Kai Ulthofer, who is also an excellent researcher, background in physics and neuroscience, machine learning, and everything else, the kitchen sink, who has written this little piece called Life and the Second Law, where he again goes through these the same framework in a very intuitive way, driven by examples, without dumbing it down. It's also highly recommended. Then finally, there's a more long-form video by Maxwell Ramstad, who is also a researcher, heavily focused on this multi-scale idea, called A Tutorial on Active Inference, also on YouTube. The beginning of this talk was very much inspired by his video, so credit where credit is due. And then if you want to get a little more involved, here are 4 of my favorite research papers. The first one is by Christopher Buckley, who was in Sussex, I believe, who has written a mathematical review of the free energy principle. And it is probably the most meticulous walkthrough of the mathematics that underlie a lot of this. In particular, he describes in detail these sort of Bayesian thermostats, very short horizon greedy agents that we showed earlier. And you should be able to recreate that figure if you follow the recipes that he lays forth. Then there are 3 papers by Carl Friston, the grand man himself. One is A Rough Guide to the Brain, which is where this idea of the brain as basically the idea of active inference and the free energy principle was originally introduced. It's a little dated by now, but it's a really good read and presents a lot of the core ideas. Then there's one called Knowing One's Place, which deals with morphogenesis. So that models how systems of cells can self-organize to more complex, large structures, i.e. how you get something like the structure of a fetus from cells going about the business and only minimizing variational free energy at their Markov blankets. And then finally, there's one of my favorite papers called Free Energy Value and Attractors, which I've read probably 15 times now, because there's a lot of good value in there, which really deals with this idea of non-equilibrium state-to-state systems and attractive states in a very hands-on manner. And if you really want to go deep, and if you're brave, there's a 150-something page monograph written by Friston called A Free Energy Principle for a Particular Physics, where he uses these tools of Bayesian inference to derive classical mechanics and statistical mechanics and quantum mechanics. And yeah, it's a lot. It's very, very dense read, and it's a time commitment to figure out, but it is very, very much worth it. So if you're feeling brave and you want to go deep, that is the place to go. And that is my last slide. So I'm going to go back here. I'm going to stop the screen sharing, and I'm going to hand it over to Bert to sort of round off the class. And then depending on if you want, we can do like 10 minutes of Q&A or something at the end. So thank you for sticking around, and thank you for listening, and I hope you also enjoyed this as much as I did.
Okay, thanks a lot, Magnus. That was a great class, really fun also. Yeah, I want to close the class. I want to thank also all of you students for logging in, not only today, but all the previous times. It's a lot of fun also for us to, even though it's online, we had a really good time interacting with you over Piazza. Of course, we're going to keep Piazza open all the way through the exam. And so about the exam, I want to encourage you that because you can do this, this class is not about big calculations, and it's more about the idea of interpreting probability theory as beliefs that gives us permission to take probabilities over parameters and over models and over hypotheses. And with that permission, we can basically derive nearly everything in machine learning just as an application of probability theory. And the core of the exam is going to be multiple choice, so you're not going to be asked a long derivation, because then I would have to show you that derivation. You have to make a choice for one of these derivations. But just when you go through your notes or through the lecture notes, just try to follow how nearly every time everything we do is just applying some product rule. And then I think you should be fine. I think you can do it. It's an interesting topic. This, as Magnus also talked about, this idea of using probability theory to derive machine learning and even apply to life, I mean, extends very far in science nowadays. There are efforts to derive most of physics from probability theory. The book that you have as one of the handouts was from Ariel Katica. That goes a very long way in doing so. I have here a little book, it's called Natural Corporate Management. Talks about how managing companies is basically a free energy minimizing process. So you can go very far with this. Anyway, so good luck on the exam. And so we keep Piazza open. And so, yeah, so that's what I wanted to say. Magnus, do you want to keep the chat open for a while? Or, but I'm going to stop the recording and keep the chat open then. And I want to wish you, of course, all the best during the exam. So stop the recording, the chat will be open.
And thank all of you for the very nice words. It makes me very happy. So I'm going to hang around for another like 5 to 10-ish minutes and take some questions if you want.
Okay,
when we sort of run out of questions, then
I'm going to keep the record. I'm going to keep it open then, also so people can also just talk.
All right, cool. Yeah, so let's do like 10-15 minutes of open discussion, and then we're going to wrap it up. And yeah, best of luck with all your exams and everything. And I'm really happy that a lot of you seem to have appreciated it. And otherwise, as I also mentioned in the chat, if you're interested in any of the stuff, just feel free to get in touch with any of your instructors, because we're always looking for good people who are also excited about these things.
I actually had a question about the last hour or so. The data from like the rat experiment, did they ever try to model that computationally, how that would work? Could you technically model that brain function?
You could. There's a lot of work that's going into modeling neural spike trains. Sorry. Usually people use some variation of a Poisson distribution over an interval. There probably has been some modeling done on this, but in the original 97 paper, they didn't go too heavily into it because I imagine it's about 20-something years old now. However, if you're interested in that, one of the authors, Peter Dayan, has written a nice textbook called Theoretical Neuroscience, I think, which is like 800, 600 pages of like foundations of how you can go about modeling neural systems using computational models with exercise and everything. It is sort of a classic in this field if you want to get into it.
Okay, cool. And also, in the original experiment, did they also test whether they could basically uncondition the rat to go back to the original like neurospikes?
So you mean whether it tested if they could sort of unlearn the connection in the rat and just have it be at baseline at all points?
Yeah.
I don't think they did. But I imagine that since the brain is plastic and we're learning all the time, you would, given enough sort of randomness and random trials, have the rat at least in part be able to unlearn this kind of mapping. So there's work in the computational sort of simulation domain on simple agents that do that, where you have agents first learn something that's completely wrong, and then by repeatedly optimizing this functional and learning a new model of the world, they can actually more or less recover what would be a good policy or like a good way of acting. So there's some work on this in the computational domain as far as I'm aware, but I'm not too familiar with sort of unlearning in the neural domain. There's work in people, actually, where you have people have tracked the density of gray matter in motor cortex as people learn to juggle and then subsequently forgot how to juggle. And you can actually see physical changes when you measure the density of neurons. So there's some work in that vein.
Okay, cool. I was just wondering because that you could relate that to learning abilities of people and then unlearning or habits or stuff like that.
Yeah, definitely. And there's work on actually a lot of active research going into how you can model habits using this. It usually comes into having strong priors over the actions that you want to take. So when I showed off my expected free energy function, I conditioned on the use because that's a very natural way of thinking. You can optimize the use and then you can minimize your functional and then everybody's happy. But you can also like take this even further and introduce priors over the distributions over U, which will sort of, again, when you're minimizing your free energy through the complexity term, rein things in. And you can have a way of modeling habits during doing that.
I shared a link about how to learn, how somebody went through an experiment where he unlearned how to ride a bike and then tried to relearn how to ride a bike. It's pretty cool. You can see it on YouTube.
Oh, cool. Thanks.
All of the graphs about the rat that you put on were after the rat has learned the stimulus. Is there in the same paper? Is there also a graph of how the neuronal activity looks like when there's an unknown stimulus, a stimulus that's like not associated with anything positive or negative?
Not in that paper, because they focused on how basically whether the dopaminergic system encoded reward prediction errors or rewards. So that's not in that paper, but you can probably get, if you look a bit, research on how different parts of the cortex, and I'd imagine dopaminergic system is probably a good way to look, encodes just unexpected stimuli.
Because like that would be also interesting in the context, because if that response is just a response to now we have to like remodel our build of the world, or if it's a response to like we have to remodel the build of our world according to like a positive thing, like I think that would also make like a difference on how to interpret it if that the same response happens to like a neutral stimulus.
I mean, whether the valence of the response has some influence on how it's encoded. So, like, if it's a positive stimulus, it's encoded different from a negative stimulus. Is that sort of what you're getting at?
What the paper says is that like the activity is because the rat adjusts like the model of the world according to what it
It's because
sees,
the
right?
stimulus that rat perceives is not predicted by the model. So there's a prediction error, and that prediction error is encoded in dopaminergic spikes.
Yeah. So if something that happens has no association with anything positive happen, that will still be something that's not predicted by the model. So
so the prediction would be that there would be a spike in neural activity.
So it would be interesting to see if there's like a difference between that spike and the spike of the reward, wouldn't it?
That would be kind of cool. I think you should get some rats and some funding. But yeah, that would be kind of cool. I'm not aware whether that has been tested, but maybe it has. If
sounds
that.
Like something that has been tested, doesn't it?
It does, but I'm not aware of any sort of specific papers testing it. So maybe you can find one. Yeah. If not, then I think you should get some rats and some funding. All right. I think we are down to like 2 or 3 people here. So I am going to call off the session. Thanks for sticking around, and best of luck.
Bye-bye, everybody.
Thank you for the lecture.
Well, thank you for being interested. I'll see you sometime, potentially.